Good afternoon, everyone. Thank you very much for letting me speak here. My name is Zheng. I am a PhD candidate from the University of British Columbia, Vancouver, Canada. Today, I'm going to talk about human atopic diseases on a chip by developing an ex vivo drug discovery platform. Okay, in this study, we mainly focus on atopic dermatitis. So AD often develop early in childhood and is characterized by, as you can see, by this extensive scaling and itching. AD is one of the most common inflammatory skin diseases affecting about 25% of children and 1 to 3% of adults. Meanwhile, AD doesn't just show up by itself. More than 50% of AD patients undergo atopic march. Atopic march means that AD actually progresses over time. Patients with AD, they usually develop AD after birth and followed with food allergy, allergic asthma, and eventually allergic rhinitis. This natural progression of atopic diseases are called atopic march. Patients with moderate to severe AD is even more likely to develop this progression. And recent definition has been revisited that if you develop more than one condition after AD, it is considered atopic march. The possible reason for atopic march could be including skin barrier impairment, genetic predisposition, environmental stress, and immunological dysfunctions. Current treatment methods have a dose-limiting effect and do not prevent the atopic march. So therefore, is really a big need to develop a novel mechanism or a medication to target this key player of atopic dermatitis in general. And thus, this will also provide a preventative measures for atopic march. And more importantly, following with the development of this drug, we need a complex human-based modeling system and to test the treatment's efficacy and safety. So later on, we will jump into a organ-on-a-chip modeling system later in this presentation. To address this, the key component is called thymic stromal lymphopoietin, the TSLP. It is a keratinocyte-derived pro-inflammatory cytokine. It is actually considered the key regulator to manifest atopic march by activating all these different kinds of immune cells and facilitate with secretion of inflammatory cytokines. They will also actually exert certain activating pathways and to exacerbate the atopic symptoms. Considering the limitation of current treatment, including the side effects and mode of administration, we therefore looking to develop a topical, non-invasive approach of small molecule drug to regulate TSLP function and potentially treat atopic diseases. So here we actually show you the ternary complex of TSLP. How does it work? So first of all, sorry, I can't find it. TSLP will bind to its receptor TSLPR to form the binary complex, and this binary complex will recruit IL-7R alpha. So this entire shape will actually activate certain signaling pathway to exert expression of cytokines. And then what we did was that we actually used a commercial database and performed virtual screening for over 1.5 million compounds. And later on, because we want to fit those groups in the binding sites. And then later on, we performed the structural filtering and docking. So eventually, we narrowed it down to a 12 virtual screening hits. So with that, many numbers, we finally selected compounds that downregulate the expression of TSLP-mediated cytokine interleukin-4 and interleukin-13. Building on top of the structure of those 12 compounds, we actually synthesized some new compounds, from which we identify the BP79 here having the most inhibitory effect for the cytokine expression. And this could potentially manage the atopic symptoms. Later on, we actually identified a concentration or dosage used in the micromolar range, which is really low compared to the current treatment method. Later on, we want to investigate the certain activated pathway that have been affected with TSLP. So here it is actually, we see with the Western blot, we see the phosphorylation of STAT6 and STAT3 in primary CD4 T cells and primary keratinocyte. We see that with the application of BP79, they actually abolish these two pathways to prevent the cytokine expression. And with all these things, we see, okay, we validated that it's working, but then how does it work? Here, we want to see if TSLP actually do the job as we propose like it disrupts the ternary complex. So we perform this proximity ligation assay to visualize the TSLP-R and IL-7R alpha's combination, or their co-localization and expression of the complex formation. So here we see that in the TSLP BP79 group, there's no ligation with the specific antibody, while the TSLP only group, we show there is the ternary complex still there. So which means we actually propose the right thing. It actually disrupts the ternary complex. So later on, now we talk about, because we established the BP79 as a potent inhibitor of the interaction between TSLP and its receptor, we need to validate this in a humanized AD progressional model. So current model we use, you can see, are either the transgenic mouse model that have the spontaneous AD-like lesion, or the epicutaneously sensitized mouse model has the artificially elicited AD lesion. So those two models are widely used. But although they're both widely acknowledged, there are still some flaws that prevent them from the appropriate candidates for AD testing, AD drug testing. Why is that? So first of all, let's see the anatomy of different skins from mouse and human. We see mouse have a relatively loose skin attachment compared to human, and it displays with a heavier hair coating and a thicker epidermal layer compared to that of human. So that actually provides a very different drug testing platform for a certain drug. And later on, considering the healing mechanism, for mouse, they heal by contraction. Human, we actually heal by reepithelialization. So those differences are really prominent, and it will definitely affect the drug performance. So here, if we want to go further into the atopic March model, it's a little bit different. So first, the thing what we do is we epicutaneously elicit the animal like AD-like lesion, and then further intranasally induct the asthma symptoms. So this whole animal model takes really long to establish, about 50 days. So that's the one reason. But the more critical reason is that most rodents, they do not develop atopic diseases spontaneously. They don't do that. It's very different from what humans have been seen. And also, there are very poor functional analogy between human and murine TSLP, with the fact that the skin anatomical differences are very prominent. So here we don't consider this 50 days models to be the one we are going to use, although it is still very widely acknowledged. We propose something different. So here we actually produce our own skin disease model by seeding a collagen-embedded primary human fibroblast into a trans well, and then seed the filaggrin knockdown keratinocyte on top of it. So filaggrin is actually responsible for the epidermal integrity of human skin, and that's how we include it there. And then after seeding on top of it, we raise the trans well into an air-liquid interface and cultivate them for 2 weeks before harvesting. To further establish the disease condition, we also add activated CD4 T cells and the certain inflammatory cytokines interleukin-4 and 13 in there to exacerbate the symptom. So overall, by doing this, we establish our own diseased skin model. So to further recapitulate human atopic March condition, we develop a microfluidic 2-organ chip setups to study the efficacy and safety of BP79 in a complex human-based modeling system. So this organ-on-a-chip platform contained an atopic-like skin disease model, and then a healthy lung model, which is a bronchial epithelium, and then with the circulation of activated CD4 T cells in the bottom. And to note that the media is actually flowing from the diseased skin model to the lung model. So this is actually what happens in vivo. To ensure that, the activated CD4 T cells, the reason we use that because it's actually the most critical effector cells of the biomarker TSLP that we're mentioning above. And our lead compound, which is BP79, was topically administered to the skin model, and then the flow underneath the model were mimicking human physiological condition. And then, so with this system, we actually hypothesized that this organ-on-a-chip modeling system could translate the drug efficacy from here to that in human condition. In general, what I did, it looks a little busy, but I'll explain. So first 72 hours, we prepare the chips by maintaining its physiological temperature and perfusion rate. And after that, we transfer the skin and lung model into the chip and start the experiment. So on day 1 and day 2, we include the activated CD4 T cells in there to mimic the disease condition. And on each day, day 1 to day 4, we apply the drug BP79 once per day. And on day 2 and day 4, we collect the media for further analysis. And as a control, we use a clinically approved calcineurin inhibitor, tacrolimus, also in here, and the treatment method is the same as BP79. We think it's an adequate control because it has a completely distinct mode of action compared to BP79, and it's already proved working. And at the end of the experiment, we performed RNA isolation for the skin and lung model. We performed the LDH and glucose quantification to ensure the safety and correct glucose metabolism. We performed ELISA to measure cytokines level and H&E staining, immunofluorescence staining to see if there's any biomarker changes or morphological changes. So here you see like the lung models here. So till now we focus on model establishment and validation. So from the H&E, no histological, sorry, this laser thing doesn't, okay. So the lung histological model here, it doesn't really change when co-culturing with a healthy control. But however, when it actually co-cultivated with the atopic condition models, at least for the skin, we see like thickened epithermal layer and extensive parakeratosis and certain keratinocyte invagination. While the lung model also shows some differences as it loses its, it has impaired epithelium with these cohesiveness loss, and then it has partial cilia loss, and it also has a pronounced cell shedding with cell hyperproliferation. But however, when it's treated with BP79 and the tacrolimus, we see the keratinocyte invagination has not disappeared, but for tacrolimus it disappeared, but for the BP79 it significantly reduced. While the lung model actually regained its cohesiveness, and then we don't see that cell hyperproliferation anymore. So similar things that we see in BP79 and tacrolimus. So that being said, we can say that at least at this part of the study, we see that BP79 demonstrated similar therapeutic effect as tacrolimus. So later, we performed the LDH quantification and glucose quantification. So this is a validation of this model. And for LDH, what we did is that we see in day 2 and day 4, like all the way through here, the LDH level is in line with that of tacrolimus. And they has not significantly changed the microenvironment of the modeling system. The second parameter, the glucose, similarly, the level is very similar to tacrolimus in terms of glucose consumption. So we say there's no significant change in the glucose metabolism. Except for the fluctuation during media change, we can say the LDH and glucose level remain constant, and is indicative we have a homeostatic modeling system set up. And this is our ELISA data to test the media in the chip. So what we did is like we see there is a high level of classic inflammatory cytokine interleukin 4 and 13 in here, so up in the disease model. So that is clear already. So after treatment, and then we see a distinct reverse of the biomarker TSLP and periostin. So they also being increased, which means the condition has been reversed. And moreover, we see after treatment, the interleukin 4 and interleukin 13 level, they go in line with the, at least for actually being reduced. And then for TSLP and periostin, we see the level is quite low over here. And surprisingly, or not so surprisingly, we see that after BP79 treated group, they actually going back to what we see in the healthy treatment group, so which is nice. And then in the tacrolimus group, we actually don't see that, which is totally expected because the mode of action are completely different for BP79. In general, we can see that compared to tacrolimus, BP79 treatment downregulate the markers IL-4, 13, TSLP, and periostin, which is responsible for the itching. So it actually decreased itching, which is great. Compared to that, so the control is a filigree knockdown diseased model we just established. So BP79 group compared to the control group, we see there's a restored with the impaired epithelium by reversing the filigree expression. While we see in the tacrolimus group, we don't see that due to the mode of action, they're completely different. So here we see with the BP79 treatment, we restore the impaired skin barrier by upregulating filaggrin expression, or say reversing the filaggrin expression. And here we also see in TSLP expression and CD4 infiltration are very prominent in this atopic-like skin disease model. And in the healthy skin model, we don't see that. So after treatment with BP79, we see the decrease of TSLP expression and decreased CD4 T cells infiltration. While in tacrolimus, which agree with the ELISA data, we don't see that changes. Although the CD4 T cell infiltration has been improved, but not as much as BP79. So here we can say that compared to tacrolimus, BP79 downregulate TSLP expression and downregulate T cell infiltration within the skin. Talking so much about therapeutic effect in skin, I want to reiterate that the idea of atopic motch, it is actually a disease progress from AD to other symptoms. So here we still want to talk about the lung condition, because the lung condition is also critical explaining how well the treatment works, how the treatment effects looks. So for the bronchial epithelium cells model here, we see increased TSLP expression in the untreated settings, and then the T cells infiltration. And also after the treatment of BP79, we see gigantic reduce of the TSLP expression and also less pronounced CD4 T cell infiltration. Although this is not as pronounced as that in skin, but still we see there's a trend of the therapeutic effect. Similarly, and then we see this, so overall, we can say the atopic motch, we propose like there is a skin and lung crosstalk between them. So from this data, although the BP79 is actually administered on skin, not on the lung, we can say that skin actually conveys signal systemically to the lung. And then BP79 treatment is exerting a positive therapeutic effects to both organs, so which is really promising to treat atopic motch. So then we want to gain additional insights on the effect of BP79 treatment on both skin and lung tissue, and we subject them to bulk RNA sequencing, especially on the lung. When we compare them, we see in the gene level, there's pathway enrichment analysis for the KEG pathway and the reactome pathway. So those are really important data because it demonstrated beneficial effects on the lung tissue because they're responsible for immune regulation, like the chemoattractant for leukocyte, activation, maturation of the immune cells. So all that beneficial effects we were seeing on the BP79 treated lung model. And to be specific, the sodium proton exchange pathway is actually responsible for the water regulation in the lung fluid, which is also displaying a beneficial effect to the lung. And in general, we can say that BP79 exerts a potentially protective effect on the lung epithelium. So talking about all that, so our BP79 have positive therapeutic effects on both skin and lung. So it is actually a preliminary, or say starting point of saying it is actually treating the atopic motch. So in conclusion, this study demonstrated a complex human-based drug discovery organ-on-a-chip platform for atopic diseases that might close the gap or of the translational study of this disease. And to the best of our knowledge, the drug we proposed with this OOC system, we proposed the first report of potent inhibitor of TSLPR that can potentially prevent and treat atopic diseases. And it will also serve as a starting point for further development in targeting TSLP as a central player of inflammation. At the end of the presentation, I would really like to acknowledge my PI, Dr. Sarah Hetlich, my colleagues and my collaborators, and I genuinely appreciate the travel support I received from the MPS award, and then the Bajang Ackwell Memorial Foundation. This really makes this trip happen. I genuinely appreciate it. Yeah, thank you.
Thank you for that talk. Very interesting stuff. Does anybody have any questions?
Great presentation, thanks. I was wondering about how you, in the end, decided the dosing for the BP79 in your model, in your co-culture or two-organ model? Because yeah, it may be difficult, of course, to without having any pharmacokinetic data or to know what to actually set as the expected level also for future in vivo translation.
Yeah, that's actually one limitations of our study. So in this slide, in panel B, we actually did the dosage testing, and then we identified 10 and 20 in there, but so far that is the data we see. And we also use this 20 micromolar dosage to our chip system to test the efficacy. But yeah, you're right. We actually should do a PK or dynamic study to further validate this value.
And in terms of future dosing, would it be applied topically on the skin, or would you have to take this orally, or how is this?
On the skin. We want to use it as a non-approach topical application of the drugs.
And just out of curiosity, how did you then apply that in the skin model? Did you make it oily and actually rub it on your skin model, or?
We actually dissolve it in the DMSO, which is a vehicle control in there, and then apply to the skin, disease skins model and let it, but in a very small volume, and then we let it drain there. The other point we have this study here is because the other treatment, their dosing are pretty high, so here we actually focus on a micromolar range, which is like way less compared to other treatment and might exert less side effects. That's what we proposed.
Great, thanks.
Any further questions?
Yeah, thank you. Very, very interesting. Very good talk. I also have a question regarding the application. Maybe I missed it a bit. So how do you apply then, the air-liquid interface? Because that would be a spray, right? That would be treating the lung.
We actually didn't use because we use this tissue use chip. We have the skin model in this air breathing compartment and the lung here. So basically what we did, we just opened the lid and then apply the DMSO dissolved BP79 on top of it with a very small volume.
Liquid.
Lifted.
Okay, but then
because in this chip, we actually can reach an air-liquid interface considering the small amount of media being in there.
Yeah, because in practice it would be a spray then for the patients, right?
We haven't gone that far yet, so right now we probably want to make it into a cream other than a spray. So that's what we propose. Because in this, give me a sec. So if with this condition we consider using it as a cream would be more acceptable for the babies.
But then for the lungs, if you would treat the lungs?
We wouldn't treat the lung because
okay.
there's a atopic match is skin to lung, they crosstalk. So we hypothesize that by treating the skin, we already see some therapeutic effect in lung.
Okay.
Yeah.
Thanks.
I think we have time for one more. Okay, we can thank our speaker one last time. Thank you.